Hello everyone, welcome to Unboxing Biology with Smriti. In the previous video, we have discussed about monohybrid, dihybrid and trihybrid crosses. We also did examples of that. In this video, we will be discussing about some types of cross which has been given by Mendel. So types of crosses. Okay, so there are mainly three crosses. First one is test cross. This is a test cross. Second is back cross. And the third one is reciprocal cross. Okay. So now at first we will be discussing about the test cross. So to understand test cross, I'll tell you one example. It will be easy to understand. If you see a tall plant, okay, now you know the organism phenotype. Okay, phenotype means physical appearance. You're knowing that it is tall in nature, but you don't know its genotype. It can be heterozygous tall or homozygous tall, but this is unknown. Fine. So to find the genotype of the unknown, we do test cross. Okay. So now the main aim to do test cross is that to identify the unknown genotype with the dominant phenotype. Okay. Or you can say to identify the unknown genotype of any organism having or showing the dominant phenotype. Okay. So how we identify this? We just see the ratios. I'll be telling you how. First, there are two possibilities, or the two possible genotypes can be there of that tall plant. Two possible genotypes. One is it can be homozygous tall. Fine. Second is it can be heterozygous tall. Okay. Heterozygous tall. So what we do, we just cross the unknown genotype with the homozygous recessive of the same character. Okay. So what we do in the test cross, we just cross between the unknown genotype with the homozygous recessive of the same character. Fine. So we will take examples. First can be capital T, which means it can be homozygous. So what we do here, we do crossing between the homozygous dominant or the unknown genotype, you can say. Here we are just taking the possibility that it can be homozygous also. So first we take this and cross with the homozygous recessive of the same character. So this one is dwarf and this one is tall. Okay, so if we do crossing, the two gametes are capital T here and the small t here. The next step, let me write first here. These are the gametes. This is acting as a parent. Now, after this, we establish or make Punnett square. Fine. So when you make a Punnett square, we will take the two gametes. Okay. Now we will write this is male. You can write over here also. This is male and this is female. Okay. So now just make a Punnett square showing male and female. Here it will be capital T. Here it will be small and small. Fine. So when we cross, we get capital T, again capital T, again capital T, and the same in the last also. Fine. So after making the Punnett square, we just take out the phenotypic and genotypic ratio. Okay. So here we just have to take out the phenotypic ratio. The ratio here will be 4:0, where 4 is tall, whereas 0 is dwarf in nature. Fine. So if the ratio is given, that the ratio, there is an organism which is tall in nature, okay, and there is a ratio given 4:0, and you have to find the genotype, then you have to do test cross, and when you do test cross, you take this possible output and then you do or make the Punnett square. You got the same ratio which is given in the question, so you will tell that the unknown genotype is homozygous in nature, or the homozygous dominant in nature. Fine. So now we will take the second possibility. So second possibility is it can be heterozygous tall. Fine. Heterozygous tall. Now we have to cross this heterozygous tall with the homozygous recessive one. Fine. Because we do crossing between the unknown genotype with the homozygous recessive form of the same character. Fine. Again, we will get the gametes as capital T and small t. Here we will get small t and small t. Okay. So now we will establish a Punnett square again. Okay. Now we will write the gametes. First write here parents, then gametes, then this is the F1 generation of the Punnett square we are making over here. So again the male and female, we are writing the genotypes over here, or the gametes over here. Now we are crossing between these two gametes. Capital T small t will give this, capital T small t, this will give small t, and again small t. So now here the phenotypic ratio which we are getting is 2:2, because 2 of the genotype is tall in nature, whereas 2 is dwarf in nature. So we can write this 2:2 as 1:1. So in the question, if it is given, the ratio is 1:1 and you have to find the unknown genotype of any organism. So you will do again the test cross and you will take the possible genotypes. And if we are getting this ratio, it means your genotype or the unknown genotype is this. Fine. Now the second one is back cross. Fine. So if you want to improve your breed, improve the breed of any organism, or you want the character of the parent must be present in the offspring also, then you can do this back cross. So to improve the breed of an organism, or you can say the offspring, you have to do the back cross. What you do in the back cross, you just cross with the genotype with homozygous dominant. Okay, in case of test cross, we were crossing with a homozygous recessive, but in the back cross, we will cross with a homozygous dominant form. Fine. So to improve the breed, it will be either heterozygous, two possibilities again. It will be either heterozygous, which we want to convert into the pure dominant form, or the recessive form. So there are two possibilities which we want to improve, okay, or the two breeds which we want to improve. So what we will do, we will just cross with the homozygous form. So now capital T small t, that is the heterozygous form, and we want to improve the breed, we will just cross it with the homozygous dominant form. Fine. Homozygous dominant, this is heterozygous. Okay. Now again the gametes will be produced as capital T small t. Now here capital T capital T. Fine. Again we will make Punnett square. Okay. As a male and again a female, right here gametes. This is parents. Now this is capital T small t. Now capital T capital T. You will get capital T capital T. This will be capital T small t, capital T and capital T small t. So in this generation you are getting every or each genotype as tall. Fine. So you are improving your breed over here. Now if we take example of the second breed, that is having the genotype small t, or the second organism having the genotype small t. Now again improving the breed by crossing it with homozygous dominant form. Again the gametes will be capital T here, and here will be small and small. Fine. Now this is acting as a parent. Now these are the gametes. Okay. Now you have to establish a Punnett square. Okay. Wait. So now you just have to write the gametes over here. First, let me change the color. So this is the male and this is the female one. So capital T, small t. Now again when you will cross this recessive form with the dominant form, you will again get all the genotypes, or all the offspring produced over here are again tall in nature. Fine. So you're improving your breed over here. Fine. In the back cross. Now the third one is reciprocal cross. Fine. Here what we do, fine, what we do over here, let us take example A and B as a parent. Here just in case, take it is acting as a male, or it is a male. Fine. A is male, B is female. We just take out the gametes and we cross between them, offspring is produced. Fine. Now in the second crossing, the A we will change to female and the B is the male one. Again we will do the crossing. What happens in this case? Why we do reciprocal cross? Just to check that the parental sex is independently crossing or not. Crossing or not. That is, the crossing is independent to parental sex or not. We just check over here. Fine. So now there are three types of crosses. We will revise. Fine. There are three types of crosses, that is test cross, back cross and reciprocal crosses. Fine. Now at first we will discuss the test cross. Here just what we do, here we do the test cross just to identify the unknown genotype of any organism which is having the dominant phenotype. Okay, now the two possible genotypes, if you see or a tall plant, there can be two possible genotypes of that organism. It can be homozygous or it can be heterozygous. Okay, now what we do, the homozygous, we just cross between the unknown genotypes with the homozygous recessive of the same character. Now we just took the two possible genotypes or the two unknown genotypes, what we have just thought that it can be the two possible genotypes. It can be homozygous also, it can be heterozygous also. So we did the cross and we got the ratio as 4:0, and in the second we got the ratio as 2:2, that is simplified to 1:1. So in the question, if it is given that you have an organism or you have a you have a tall plant, fine, and the ratio of it is coming 1:1, the phenotypic ratio is coming 1:1, and you have to take out the unknown genotype, or you have to take out the genotype. So here the genotype is unknown. So again we will do the test cross and find that what genotype is there. Fine. Now in the back cross, why we do back cross? Just to improve the breed of an organism. Here the organism genotype is crossed with a homozygous dominant. In the test cross we did crossing between the unknown genotype with the homozygous recessive, but in this case we are doing with the homozygous dominant. Okay, now again the possible, it can be heterozygous or it can be recessive form that we want to improve. So now we did again the crossing between the heterozygous with the homozygous dominant just to improve the breed. So now here in all the cases we are getting the good genotypes, or with the tall or the dominant genotype. Fine. Now here also again we crossed it with the homozygous dominant and we got this. Fine. You can write here the ratios also. The phenotypic ratio. After making Punnett square, you have to write the phenotypic ratio or the genotypic ratio. It is 2:2. Okay. Sorry. This is 4:0. I'm sorry. 4. Okay. So 4:0. 4 are tall, whereas 0 is dwarf. Fine. Now in the second also, the phenotypic ratio here will be 4:0 again. 4 is tall, whereas 0 is dwarf. Fine. Now in the reciprocal cross, we do nothing. We just exchange the sex, you can say, where here we are taking the two organisms A and B. Now A is acting as a male and B is acting as female, and we do cross between the gametes. And again in the next step, what we do, we just exchange their sex. A becomes female and B becomes male, and we again do the crossing. What happens by doing this? Why it is important for us? Just so that we can know that the cross is independent of the parental sex or not. Fine. So in the next video, we will be discussing about the incomplete dominance. Thank you for watching my video.